Hello, and welcome to week 6 of our 6-part, of course, right? Machine learning workshop. Thank you for joining us through all of these. If you made it to the end here, you've completed the entire course from basic R to basic statistics to more advanced machine learning. So it's great to have you here at the end here. Today we're going to be talking about, it's more conceptual here. We're doing kind of a transition lecture here, a transition workshop, to where, kind of like when we ended statistics, we did linear models to try to bridge over to machine learning. Well, now we're going to do neural networks, because guess what? Our next series is going to be on deep learning. Well, let's do this. So week 6, neural networks and transformers in cancer research. So today, don't worry so much about the code, exactly what's happening. Just want you to big picture to see what's going on. Because a lot of this stuff we're going to later do in Python anyway, so it's no big deal. However, if you want to learn more in depth, dive in, look at the code, copy the code, play with it. I definitely encourage you to do that. I'm just saying that I don't expect you to completely know all of this stuff. This is the introduction to this, right? This is a very advanced topic. All right, let's get into this. So last week, we explored, the traditional machine learning classification methods, right? We did regression, more continuous classification, and then we did, categorical classification, right? So we looked at methods like logistic regression, random forest, XGBoost, and we saw things like random forest and XGBoost just really, really great with tabular data, right? However, they're really best in class, really. In industry or in genomics, these are the best in class algorithms for tabular data. However, they have limitations dealing with other kinds of data, like images, like slides, sequences, DNA, RNA sequences, longitudinal patient data, looking kind of networks, gene regulatory networks, the protein-protein interaction, and then even text notes like medical clinician, notes, things like that. So you can see that there's a lot of other kind of more interesting data that neural networks can be applied to, and really, they're perfectly designed to handle these types of data. And really, they form the foundation of deep learning, which has really revolutionized everything. We all know all the craziness AI right now, right? Well, it's going to get more and more and more, and I hope someone watches this video 5 years down the road and is like, oh my God, now it's here. It's doing everything. Anyway, so this week, we're going to talk about the relationship between machine learning and deep learning. This is what I really care about you really, really paying attention to today, is explaining the core neural network concepts, okay? And things like the neurons, layers, activation functions, backpropagation, how do these things work conceptually, right? And then we're going to build and train a few methods and tidy models and using, Torch in R. Torch is mainly used in Python, but this gives us an example to end our series in R and kind of look over those things. And we're going to go through things like the multi-layer perceptron, convolutional neural networks, recurrent neural networks, and a little intro into things like graph neural networks and transformers. And you know how big a deal those are right now. Everyone's just using those for everything, right? And we'll recognize the limitations of R too, if we're dealing with things like GNN and transformers, and when you want to really move over to Python to do this. And we'll kind of compare just in general how they fit in with machine learning methods. So here today are all the required libraries we're working on today. A lot of these are very similar that we've used before. The bottom here is just all our data sets. I would look at these for parallel processing. We're using these guys. But really today, these are the new ones here: Torch, Luz, Brule, and Torch Vision. And let's just be able to Brule does tidy models, but the other three are bringing in Torch into R to be able to use. Now you can access Python in R through things like reticulate and other methods, and we could have run that way, but it's really buggy and it doesn't really work all that great, and I don't recommend it. So if you want to do things in Python, do it in Python, not in R, right? Anyway, but this is just good for our kind of introduction and transition here. So first, what the heck is deep learning anyway? Well, if you ask different people, they'll tell you different things. But I think a really easy way of putting this is you have artificial intelligence, AI, and pretty much everyone just calls everything AI. But AI is probably just a broad category that contains all of these two methods, machine learning, deep method, all of these things where a computer, these computers are solving big data. Really, that's artificial intelligence. Honestly, even Microsoft Excel is artificial intelligence. Anyway, it's a super broad category, right? And then you go to machine learning, kind of the things we've been dealing with the last few weeks with our random forest, XGBoost, linear and logistic regression, things like that. I consider those machine learning, which is a subset of AI, right? And really, it's pretty obvious. What is machine learning? A machine is learning, right? So we're giving it data and it's learning on the data and going through it in different kind of ways and learning from the data. And it gets, as it moves through the data, it learns more and more and it provides us our solution, right? Instead of just having something that like statistical method where it's just a one shot, there's no learning. There's just a one shot for most of them, there's only a one shot classification or execution of the algorithm, right? It doesn't learn as it goes. So that's the whole concept of machine learning and why it's so powerful. That's why we have all these data centers being built, all these things. You don't need data centers for linear regression. Anyway, so the next step is deep learning. So I was considering deep learning another subset of machine learning, right? And here, I used to say deep learning is image data, and machine learning is tab data, but now they're both, there's a blur of the line between the two. So I think it's best just to consider deep learning as when you start bringing in the neural networks. When you start bringing in neurons and stacking neurons, having hidden layers, all sorts of things like that, that's moving us toward, deep learning. And here's just kind of an explanation of these right here. And you'll see things like machine learning, really, really good at tabular data. And where deep learning is really better at images, sequence, graphics, and text. But that's pretty much the summary of this, but you can look into a little bit more. And let's just go to the next step here. This is, I thought, a really cool explanation of looking at a neural network from a very beginning perspective. So what is the connection between logistic regression and a neural network? Let's see. It's kind of crazy, but logistic regression is actually a neural network with 0 hidden layers. It's one neuron. Same thing exactly, right? So let's look at the little bit of the logistic regression review. If you remember, linear regression, right? We just have a combination, a linear combination of different genes or different inputs. Let's call them genes right now for X. So you have a bunch of different genes, right? And we have a linear combination of each one of them, right? Where the beta equals a coefficient to tell you how much each gene should go toward the prediction or whatever, the model. So that's linear regression, right? And that outputs like a continuous variable, continuous answer, right? And then we moved to logistic regression, if you remember, last week, and then we introduced the sigmoid function. Excuse me. And the sigmoid function, basically, you run the answer of the linear regression through the sigmoid, and it squashes it, whatever, into a probability between 0 and 1. And then that allows you to make your class probability projection or prediction, right? So that's what logistic regression is. Well, it's crazy, but a single neuron is exactly the same thing. Exactly the same thing. It's hilarious. So your inputs are your X's right here, right? No different than this. There's our genes, right? And then here's our weights. For each one of our weights to how it will contribute to the model. Well, that's the same thing as these beta things. It's literally the same thing. And then you add a B, you add an offset to it, just like this beta here, and then it's crazy. Then you do a sigmoid activation if you'd like, and bam, it's exactly the same thing. There's no difference. So just really think about this in these terms that at the very basic building blocks, it's just linear regression built up, and up, right? So this is identical to it. It's the same thing. But then what makes it a neural network then? What's the difference? Well, when we stack multiple neurons in layers, we get a neural network, right? So now look, we have our input genes. These are our X's, right? Then we have our first layer. This is a fully connected neural network with a multi-layer perceptron, fully connected. So everything is connected to each other. This gene goes into every connection. So then these guys, we'll talk about later what happens, but they look over the data, learn some new things about the genes, kind of at a high level, and then they pass on to the next layer, the next hidden layer, and they learn more complex details about the gene. So each layer is learning a different thing, but they're using the past information as a guide. And then finally, we output to two classes, like immune high, immune low, whatever. So that's a neural network, right? These stacked hidden layers that are connected, right? And like we talked about, single neuron is equivalent to log regression, right? One layer with like 10 neurons is kind of a small, basic MLP, multi-layer perceptron, right? And it starts getting you good for really very small nonlinear patterns. Then if you build them deeply, you just get more hidden layers, more neurons, and then you can deal with more complex patterns and deal with more. However, you're prone to overfitting and you need more data. That's the only thing bad about it. But we'll talk about that later. But yeah, the whole point is they become more complex as you add more layers. So how does the data flow through this? How are things calculated in this? So just kind of building on what we just showed, we have this input layer, which is like the features, the genes, right? in our instance. We have this hidden layer, which is just our different layers of stacked neurons. It's kind of the black box. We really don't know what's going on in there, which is kind of the drawback. One major drawback of neural networks is things are going on where you don't know what's happening. So this is an example, but we live with it. What are we going to do, right? So we have our hidden layer in here that has our weights. Remember, our weights are helping to help those balance those coefficients to model the data. And then we have our activation function, which produces things like letting the data go through, neurons firing, or even outputting the actual result. Then we have our output layer, which is our prediction. And then that basically, each one of our prediction gives us, we fit a loss function and we have our error for each prediction. And then on the side, we have a bias that's with the hidden layer too. That's no different than the intercept of linear regression or logistic regression. It's just basically just saying the model is not forced through the 0, 0, right? It's able to move up and down on the intercept, which gives us more flexibility, right? So that's just super, super high level what is happening in a multi-layer perceptron. So really the idea is you have this input layers are features, hidden layers that learn these representations, output layer gets our class probabilities, our prediction, right? The weights are the parameters, bias is that offset, and the activation function helps make a nonlinear transformation of the data where it can actually be more useful, right? So going to the activation function, I would consider this like the gatekeeper at the end of each neuron and our set of neurons, right? It could be either or, and it determines whether a neuron should fire. Should we pass the information on or should we just do nothing, right? It's kind of like electrical engineering when you have the 0, 1, switches, it's like, should we pass on? Which is exactly what a real neuron in the body does, right? In the brain, you have a certain signal and the signal surpasses a certain level, it activates the gene fires, right? It passes information. If you do not get above that level, what happens is different ions go across in different different cells and they create a membrane potential, right? If the potential is exceeded, the neuron fires. If it doesn't fire. So we've just kind of stole this concept from biology along with the neurals, and that's what we're doing here. That's what we're doing these functions, right? So there's 4 main functions used. The tanh is not used much, and it's more to do with certain specific functions just to improve models as they go along. ReLU is the most interesting one. It's the rectified linear unit. It's the most popular one we use, and it's used inside the hidden layers, though. So basically, if an input goes into it and it's positive, it passes it through. But if it's negative, it just goes to 0, right? So you can imagine there's 64 neurons, only certain of them are firing, right? Depending on the information they have. And that's kind of how you regulate the information, right? And so for sigmoid, it's kind of like we talked about, it's squishing to the 0 to 1. So this is more of an output, squishing into a probability of 0 to 1 for binary classes. Or softmax is the same thing as sigmoid, except we don't have binary classes here. We have multi-class results, right? So it basically gives each class a probability, but they all have to add to 1. And softmax is a good last output because it can work for binary or multi-class. And we'll see that moving forward here. And let's see, so here's kind of the mathematics behind just the distributions of these different guys. And the kind of the idea is you're squished into 0 to 1 here. Tan just gives flexibility for modifying hidden layers. ReLU just goes to anything. It's just firing, right? Whatever the information is. And softmax sums to 1, basically. So here we're going to learn about really, really how a neuron. So we have the kind of the overview of how it works, but what's going on in the scenes to really update and improve these models. and how do they learn here, right? So there's this thing called forward and backward pass. Every time data goes into a neural network, it runs a forward and a backward pass. So the forward pass is actually making our prediction. The backward pass is learning from our mistakes, updating our model, and epics are just repeating that over and over and over again. That's really all it is. So in our case, we have our input genes. We put them into, for example, we have a 2-layer MLP here. The first one, we basically, you see, let's just say we have 64 neurons here, 32 here. It doesn't matter. This is a teaching example. But imagine our first hidden layer, we have the weights and our offset. So we set the model, we fit the model, right? And then we run our ReLU activation functions. Certain number of our neurons actually fire, push on the information. The second hidden layer of neurons grab the information. The hidden layer is probably looking for one thing. Hidden layer is kind of building upon that, looking for something new, are looking more in detail. Either way, and it fits the model again, runs ReLU again, shoots off the information to softmax, and softmax does the activation function that creates the probability, right? So now we have an answer. We have our first prediction. So now, how do we do? Well, we need to compare back to the true label to get that, right? So then we compute the loss function and compare back to the prediction, to the true label, and see our error, right? And then once we have our error, we're like, what can we do to make this better? So which weights cause the error, okay? So we basically do a gradient descent. So gradient descent is, you're basically, imagine you're hiking and you're trying to get out of, you're stuck. So you're kind of just walk around different directions, and as you walk, certain areas get higher, certain areas get lower, and you try to kind of just walk your best way to move up to the top and get the highest level. And that's kind of what gradient descent is. It's just kind of a walk through a random walk that keeps trying to get to a better spot. Now, what can happen bad is it can get stuck in a valley sometimes, so we won't go there. That's basically how it is. It's walking around, trying different things, and until it finds a better representation of the weights to fit the model better, right? Based on this loss. Then it updates all the weights, and then it shoots it back to the original genes, and then we put it in again with our new weights. And then we do it again and again and again and again, each time at epic. And then then we get our final outmax and our softmax. And then based on the loss, we choose which one we like the best. And that's a super, super simplistic way of thinking about how we end there. But the idea is that to improve the loss, get the lowest loss, right? So once again, the forward pass, the data is going in the network. Each layer is computing an output, and then we give a prediction. And then the backward pass, we're learning from that, right? We compare the prediction using the loss function. We reset the weights, using gradient descent, and then we update them, and then we just keep repeating until we feel like we have a good answer. So that's really, honestly, that's it. There's nothing more. Based on what I showed you before in the single neuron and that is what a neural network is at its core. And for the loss function, we won't jump into the math here because it's kind of complicated, but we usually use the binary cross function if you have a binary result, or the categorical cross entropy for a multi-class classification. But don't worry about this today. I think the best way of thinking about this is our binary cross entropy here is very similar to our logistic one, as we would expect, right? The idea here is just you apply the different loss functions. You can look at the math later if you want. I don't want to drown you in that. Okay, so there's our background. Now let's create some data sets and run some models. So first I want to create 3 data sets that we're using today. The first one is going to be a tabular gene expression for the MLP. And for this, we're just going to keep continuing what we did the last 2 weeks. I'm using this BRCA data set, right? This breast cancer data set that, and we're going to grab about 500 cases this time. And we're just doing the old GDC query like we all have been the last few weeks. So we're just using that same expression data, grabbing 500 samples, doing all our VST transformation, just like always, grabbing the top 500 most variable genes, centered scaling it. One thing that I'm using Excel again, the Excel analysis. If you remember from last week, that gives us a lot of, it uses signatures to find different things like macrophage concentration for M1, M2, immune high versus immune low, blah, blah, lots of different things. So just building upon that, we're using the same one as last week, so we can kind of compare it to it. So basically, I just found that. I found the immune score, found the median, and I split the data into the response into immune high versus immune low, exactly like last week. So we just have expression data, tabular data, immune high versus immune low, binary classification problem. The second data set, which we're going to be using for the convolutional neural networks, so we need image data, right? So I think I made some pretty cool data here. I basically programmed this myself and created some synthetic data, some synthetic radiology data. And the idea is here, we're looking for tumor images that have a certain architecture, that have a certain brightness in the center, a denseness, a certain topology and size. And basically you can see kind of here how this is separating the tumor from the normal, and our model is going to jump in there and try to predict this based on the data. So we'll be looking at like radiology sample. Let's build a model if we can predict if we can find a tumor or not. Pretty cool stuff, I think. And then finally, we're going to develop some simulate some sequential data because we want to talk about things like recurrent neural networks and looking at things like longitudinal patient data. And for this one, I basically took 500 patients, and over 20 different time points, I looked at 5 features. So just think about, this is just like a cancer patient who's got a treatment, and they come 20 consecutive times, and each time they get blood work done, and each time you get blood work done, we're looking at 5 specific features. They could be the white blood cell counts, anything from LDH, your iron level. It could be anything looking toward that cancer you need. It doesn't really matter. Don't worry about so much of that today. Just think of that we have 5 features we're tracking over 20 time points over 500 patients, and can we make predictions on what happens there? So what we've done is we created 2 categories. So this is one patient. Each one of these is one patient and one marker. And kind of what we're seeing here, we're trying to look between, did you respond or did you not respond, right? And the non-responder is going to be more a relapse people, right? So if we look at responders, there's a clear decay on this marker in this patient. And even though we jump up at the end, if you look at everything, you can really tell there's a clear decay. Or the non-responder, it's not so simple. Depending on where you look, it kind of looks like there's been a response, right? But then things turn and they get worse and they kind of relapse. And it's difficult for some models to see this U-turn unless they're sequentially remembering everything has happened along every step, right? And this is really a toy example. It's easy. You might say, oh, well, this one's higher. It's obvious, right? And that's true. But remember, this is just one patient and just a few markers. Imagine a clinical trial with 10,000 patients and hundreds of markers, and they're all different. Some go up, some go down. It can be very complex to pick out something like this. So that's why you would need that model. So remember, it's just a toy example to show you how things work. But what you can also see here, these models, even though the model is trained on all the 20 weeks here, or 20 time points, that doesn't mean it can't predict earlier. So another thing is once we have models that can predict well, we can look earlier and try to catch like the momentum of this change, try to find that U-turn early on to be able to catch relapses and non-responders earlier on, right? That's kind of one of our goals in precision medicine. So anyway, that's our data set. We'll make a model later, okay? So let's go. First one is our multi-layer perceptron. It's kind of like what we've been talking about before. It's a very simple neural network with hidden layers, and it's fully connected. Remember the feed-forward architecture, every neuron in one layer is connected to every neuron on the next. And this MLP is really the foundation of modern AI. Everything's kind of built upon this architecture. It's kind of like the same thing we've been talking about, your input, your hidden layers, ReLU in the middle, softmax at the end. And we won't jump into too much of the math here, but you can kind of look at our forward pass. All we're doing is remember we talked about the WX plus B, fit the model on the weights, and then do ReLU to fire, and then softmax at the end, just like we talked about above, all while computing the loss function and going back and around, right? So we already know that. We've already gone above that. The network makes predictions. It measures how wrong they are. It figures out how to fix it, and then adjusts it to do better the next time. So how can we make one of these models in R? Well, the one thing we can do is a simple MLP with one layer. So tidy models, using the Brule package, you can make an MLP with one layer, which is just a good way to play around with it and understand what's going on, because it's the basis, right? And then you have to move on to other packages without that. So this is as far as tidy can go. But we started with tidy, let's end with tidy, right? So the same as before, we grab our expression matrix, we do that initial split, we're gonna doing 75% to the training, and then we make our training testing data, we make our recipe, we set up our model here, we're tuning some variables. The hidden units is the number of neurons in the hidden layer, the epochs is the number of time we go through, the learning rate is the step size for gradient descent, how big are our steps in that hiking trip, and our penalty is going to be our L2 regularization. So we're going to have this kind of ridge-like regularization along the way when it's refitting those weights, because it may squish some genes down close to zero because they're not useful, right? And the learning rate, think of it as the slower the learning rate, the longer it takes, but the more accurate it probably will be. Hidden units and epochs, you kind of know what I'm talking about there. And then we set the grids up here, just our ranges to do our grid search, set our workflow up, set up our cross validations, just like before. One difference here I'm doing is I'm adding parallel processing based on the package that showed you earlier. And you can always do this, just put in parallel detect cores. The cores, just set this up. You can cut and paste this code before anything that's really powerful you need more cores for. And then at the end, right after you do the fit here, so I'm doing this grid search fit here, right? Tune grid. Right after that, I just stick it back to stop cluster and go back to normal. And that's just saying, hey, use a bunch of cores, use it however many cores we want to use. I'm using like 6 or 7 cores here. I have a MacBook M1 Max. I can use about 7 cores at once, but things keep going. And so it just allows me to parallel process, run things parallel, and because these things take forever to run, even when you're doing parallel. Anyway, so anytime you want to do parallel processing, you put that in the front, that behind, and then go back to normal. And when we ran that, we saw here, looking at this mean column, I looked at it's the AUC. You see here the metric, the mean, and the best one was here. And we see here kind of the parameters that go along the best with that model. And here's kind of an example. If you want to look into this a little bit more, it's just showing basically, you look at the ROC here, it's basically looking at the epoch and the learning rate, amount of regularization, the hidden units, and it's kind of trying to look at all of that at once. And this is where we drew, our best model from, if you want to take a look at that later. And we kind of went over this earlier, if you want to look more into the parameters and the typical ranges you would find these things in. So we selected the best model based on our select best, just like normal, and did our training just like before, finalize workflow, fit, and get predictions exactly like the last 2 weeks. No difference. Only really difference is we're getting different metrics. We want a little bit of difference here. We have it's just really accuracy, CFP, and CAP, pretty much the same. So let's look at that, and then really got to focus more on the accuracy and the ROC, and we basically just ran our final predictions. This is our best MLP model on the tab data. Accuracy 78, not super great, but 85.5 ROC is actually not that bad. And this is kind of comparable to last week's. And I'll tell you, we're just trying to get the MLPs, if they can even compare and be on the same level as the random forest and elastic net, then we had a success in this lesson of showing you how to build the model. Because remember, those are the industry standards. We're not expected to beat them on these. So if the model's even close, we're success. Can I talk? So don't worry too much about, did it beat it or not? Worry about, the lesson and the architecture we're building. All right. So next step. We did the MLP with Brule, but it was super simple, right? And we didn't have much control over it at all, right? So what we can do is we can build an MLP with LUZ. And this is actually bringing in PyTorch geometric into and that's going to be what we're actually going to be using, which is the basis of PyTorch when we use it in Python. It's actually very similar, and a lot of the code looks very similar. So it's a good way to look at it here to get exposed to it, and you'll remember things later on. But this basically just gives you control of the whole deal here. So the first thing we're going to do is we're just going to make a training and test set, right? And we're basically just pulling out we're trying to organize things a little bit different than tidy, basically. Like the usual, it's a little bit more detailed. So the first thing we do is bring out just the training. So we just get rid of the the response column, right? That just gives us our test and training data without the response column. It's really not that complicated. We scaled it, but it's already scaled, so it doesn't need to be. And then we just grabbed a numeric vector, basically, of our responses, and then put those into train and test. So then we just have our train and test data, basically, of our validation, our response and expression data and predictors for the validation and training. I can't talk. So here's the difference here, and it's pretty much similar like this when you're using Torch in most of the models. Is you have to set up a tensor data set first. So the way you do this, it's really easy. You just put, for X and Y, you just put Torch tensor, your training set, and then your data type, which in that sense, float is like continuous variables, and Torch long is like factors, and that's because we have immune high low, right, as opposed to just expression numbers here. So we use that for the train and the test. We just kind of set up this Torch data set. It's basically just putting everything into tensors for us. And then the way most machine learning models work is they have data loaders. So they basically put data in batches. They don't put all the data in at once. They put it in batches. And like for instance, here we have 32 in each batch, right? So let's say we had 500 samples. They would just go 32, 32, 32, 32, and work their way through and then run through the model. And then it would just keep building upon that and learn from it as it goes. And that's just kind of how, and that's something you do. Tuning the batch size is something that is very often an important feature. But just thinking about it conceptually, don't worry about exactly why at this point. And then we have to build our model. So that's what you do in Python or in Torch. You have to build all these models. They're not necessarily given to you. So they just kind of build these functions. So for our MLP model we want to build, we kind of have this structure that this we're programming in here in the PyTorch language. So it's expecting these kind of things, right? And it uses things like these NL linear. So like I'm using so if you look here, I guess it's our input size is like our number of genes. Hidden size would be our first layer, right? It's layer 1. We're doing the layer 1. Layer 2 would be we bring the same hidden size over here, but then our hidden size we're making it half the amount. And then we're bringing that one over here, and then we're halving it again. I mean, we're bringing the same one over here. So it could be 64, 64, 32, something like that, 32 output. And our output is our classification. So we're basically just programming each layer, right? An input layer and then 2 hidden layers, and then we're adding dropout. And basically dropout is a cool thing where it basically, as you go through each layer, it randomly just turns off certain neurons. So each epoch you go through, different neurons are turned off. And basically what this is doing is it's kind of like bootstrapping. You're changing up the different sampling of the data so that the model's having to learn a more complex representation of the data. And it also keeps one neuron from talking too loud. If one neuron's yelling all the time, they can't see the others. So you might want to cut that one off one run through and see how things work out without it. So dropout rate is the percentage of neurons that are dropped out. And let's just, then we set up a forward function, and this is just putting everything through. And you see self layer 1, our first reload with dropout, self layer 2, our reload with dropout, and then self layer 3 is our output. So we're just kind of organizing that, and then we're putting it into one last deal where we actually fit it. And here, we MLP model, piped to first you do setup, and you set up your loss function, and your optimizer, which is just Atom was usually used. This is just how a function to update your weights. And then we're going to use our metrics here that we've already set up earlier. Our loss metric accuracy, it's a set up a function to get your metrics. And then we'll set our hyperparameters. And we have our input size, just our number of genes, number of columns. And let's see, our hidden size, 32 neurons for each one of them. I'll start with 32. So it'll be 32, 32, 16, 16, right? And then we have our dropout of 2, and our output size of 2 classes, and our dropout, which is pretty big. And then we'll set our epochs and our validation data, and then we just run the model. Now it's time, everything's set up now. It fit everything right here, unfitted, and based on our fitting and our test data, we can make a prediction, and look at torch argmax to get our classes and NF softmax to get our actual probabilities that actually went to those classes, right? And then we can look at our accuracy, and we see that we get 0.778, which is very similar that we saw above in the simple MLP. So we didn't gain a ton of differences with this, but we also didn't do a major tune on this either, right? We wanted to do a simple example. And then we're just using laws. Once again, we're just calculating actually we want to look at our AUC here now, and it's 0.84, which is very similar to the 0.85 above. So very similar, we expected, right? And just to kind of look at how these things train, it's often very useful to use looking at these training histories, and you can see what we're seeing here is the training and, data and our validation data. And you see here is we go up as we go to each epoch. We're learning a little bit better. We're getting a little bit better accuracy, a little bit better loss, or less loss. So you want to see similar things in the validation data, but we don't, right? It doesn't seem to be learning much as it goes along here, and the loss kind of stays the same too. So there's not much. Something's going on here where the model is likely overfit a little bit, and that's why it's not generalizing to the validation set. So we might want to go back and work on some things to improve that fit, but it actually is a good teaching example that it doesn't work, right? And so there, it'll give us a chance to make it a little better if you'd like. And let's just compare this to traditional ML methods. And I'm just going to go back and use the same parameters that we used for the same classification last week. So I'm running a random forest with our optimized parameters, just like always, and elastic net as well too. I won't go through this because it's exactly what we did last week. And then I'm kind of just adding our other data as well. And let's look at the final accuracy. So let's try to look at this mean ROC AUC. That's what we care about the most. And we see they're all very similar, right? Elastic net, random forest are very similar. If we tuned a little better, I guarantee random forest would win. And but those are very similar, powerful methods for tab data. And then we see the two MLPs aren't that far below, which is really great because that's not what they're good at. So I consider this like a success even in a toy model. And you can kind of look at the bar plot if you want, and you can kind of check out some of the takeaways later. It'll be, I just want to talk about things like they work good for tabular data, but they need to be more complex, and they really need a lot of data to look good. And in this sense, normalization is very important. It's very sensitive to initialization, and you want to use the least number of hidden layers as possible and the least number of urons. So always start simple and move up. You don't want to use a lot. Even if it's the same result, you don't want to waste all that compute power, especially going into the future where we're going to lose a lot of energy in an apocalypse dystopian world here, right? Just kidding. And then, work on fit your dropouts, 0.2 to 0.5 is a good way to do it, and realize that even then you may not be able to beat it because random forests and attributes are so good. I want to show you an example from the homework. Actually, you'll kind of see this. It's a homework problem where I basically built a similar model, and my idea was, if I just keep putting enough samples in, is it possible for the MLP to eventually pass random forest? Because that's always people argue, right? So don't worry about what's going on here. It's the same thing. We're looking at longitudinal data, and we're trying to make a prediction on it, right? And it's tabular data. And you can see here, the way this is a very difficult data I set up too. So log's having a really hard time getting better because this data is very difficult on purpose. And random forest is just taking off like a freaking race car, runs off and doing really, really well. While MLP kind of struggles at first, but then it starts getting its footing a little bit, starts catching up, and then it's really starting to take off. Look at this slope here. So if we get up to like 20, 50,000 samples, random forest is going to level off likely, and MLP is going to go up higher. So this is kind of a good example of how you could expect MLPs to work on data in comparison to things like random forest. But you can see going into the future, all our data is going to be like millions and millions of data points. So that's why neural networks are probably going to surpass these methods going forward as our compute gets better and better. Anyway, so that's a good explanation of that, I think. So then let's go and talk about our next step, which is going to be convolutional neural networks. So a convolutional neural network, CNN, is basically a type of deep learning model specifically designed to process data with this grid-like topology, right? Which is basically images, if you think about it. And while a standard MLP treats every single pixel, so let's imagine you can flatten an image, right? So you could just get the color, which will be a hex number or whatever for each pixel, right? And just flatten the image into tab data. So just each pixel will just have its color hex code or whatever, right? Or its red, green, blue number or whatever, right? And you can use the MLP on that and be like, let's make our predictions. But the problem with that is, while you would be able to have a relationship between input and output, you wouldn't know that actually all these pixels are spatially related, right? The spatial context is extremely important. They're forming an image that has detailed edges and things like that. So MLP can't even come close to a CNN this way. But how can a CNN accomplish it? How can it find these edges, and how can it do this better? And what is convolution? So the idea is things are convoluted. How can we make them more unconvoluted, more clear, right? So CNNs basically accomplish this by you have an input image and they run this slide filter. And I'll tell you, we'll show a second how that really is done, but just imagine this filter kind of running back and forth through the data, right? And while it's moving through the data, it's neurons are taking information and they're doing modeling to set up their linear combinations and all that stuff, right? So by doing this, by these neurons are all doing their thing, they're the first layer could do things like detect edges, gradients, things like that. But then so basically the idea is you run the slide over the data, you learn some stuff, then you pull the data, stick it into another layer, look for more stuff, more detailed, run the filter over it again, and then you're running as each step, the dimensions are getting smaller as well too, and then finally you flatten at the end into a fully connected neuron set up to get your output. So you're just basically learning as you go, and you stick an output. Here's, I think, so the first layer, kind of your simple run through, right? You learn your edges, maybe color gradients. I think of this as like thinking about let's think about this in digital pathology. So your first layer would be something like separating out cancer from normal, okay? And then you would be like, okay, now we know the cancer, let's look at the immune microenvironment, right? The next convolution level could be segmenting all the cells in your microenvironment, right? And the third one could be like assigning more complex, like looking at the spatial environment. Like we found the cells, like you could look at like how irregular the nuclei, things like that, or you could look at the spatial representation as well too. But the idea is, you see here, we can learn more information as we go, so we can get a final output that's really great. And I won't run through the math, kind of complicated here, so I won't run through this. Now I just want to think about this conceptually, where where a kernel is a filter. And this is really a small matrix of learnable weights. So you make a filter, for instance, we have an image here, it's 5 by 5, and we're making a filter of 3 by 3. So this is our filter, right? And it's like I said, small matrix of learnable weights. So it's fitting and learning the weights here, right? And then it's pulling them by taking, for instance, the max, it could be different things. And then all of this goes here is this -1. And then the filter slides over to the right. Now we're 3 by 3 here, if you can imagine, right? And we calculate, pull, we get the middle one here. And we just keep going through each part, right? Until we end up at the very end here, right? 3 by 3 in the bottom right corner. And then that gives you that value. So you can see how you're being able to slide over all the data and basically pull different parts, and that reduces the dimensions of the data and gives us a collective understanding of that region, right? And you can do this multiple times with the different layers, right? And you're getting smaller dimensions each time, right? And then finally you stick it into your output, right? And for example, here we could have found like there's something cool happening here, in this guy here, cell or something. And that's kind of how that works. So convolution is extracting the features, and these features learn as they go. The filters, they learn how to be better and be optimized as they go, and we can actually tune as well to find out what the actual filter should be. But there's also some obvious ones and numbers that go along with it as well, like trying to half your dimensions, things like that. like a 2x2, for example, will have it. Let's see. So, and then we do a ReLU each time to keep it going, then pull, flatten at the end, and then find your classification, right? So these are just small detectors, right? They go across the image. Each detector learns a specific pattern, something like an edge, texture, and then they pass it on, and they pull the data, pass it on to other ones, other layers, which can combine these patterns to more complex features, for example. All right, so let's build one of these in R for all things. Well, in R with torch, right? So we're using LUZ once again, and we're gonna make, we start off with torch stack here. It's another function, and it basically pulls in all these images to an image tensor. So we have an image tensor that we pulled in all the images in, and this is a very difficult processing step, to be honest, in real life. And then we set an image label tensor that's giving us, the label of what they should be, right? And then we're splitting the data here a little bit different, but we're just splitting the 0.75, 0.25, and then we're just creating the tensor data set once again, like before, and then doing our data loader once again with our batch size of 32. So you see a lot of these things are very similar, no matter what you're doing. You can see how if you learn one model, you can start learning how to do other ones as well. PyTorch has kind of a similar framework for all those things. And then we're building so this was like our data setup, and then building our actual model here, and in module again here. And then basically, this is where you change things. If you remember last time, it was like self, I forget what it was. I forget what it was. It was like self. We can look back at what it was. What the heck was it? Now I need to know, because there's really good ways of setting these things up, right? LUZ, right? Even I'm lost in all this data, right? Yeah. So sorry, layer one, right? So the idea here is I want to show you is use the NN linear here and layer one. And that's kind of how you do a normal one, a non-CNN one, where CNN is a little bit different to where you would do like, you would do convo one, kind of to keep it to where you're explaining it the correct way, and also the PyTorch understands these different things. And then we use, you can see here, NN con 2D, right? So this is, you can see the, a 2D filter basically. This is just a function. This is a PyTorch function that's helping you out with this. So one is the color you're looking for. For example, in this, 16 is the number of neurons, and 3 is the filter size, 3 by 3. So we do that. We run it to our second one. Very similar. We bring that 16 here. We jump it up to 32, and then we run another 3 by 3 filter. And then we self pull this time. So run this one, self pull, and then send it to our linear, our fully connected linear one, right? We've actually flattened it down, and then that has some dropouts. So you can kind of see here we do first layer, ReLU, pull, second layer, ReLU, pull, flatten down, run the actual result. Kind of similar to the other one, right? And then we set up our fitted thing. This is really similar. Same cross attribute, same loss function, same atomizer, same metrics. Our hyperparameters, we're just going to put 2 for the outputs, and then you see 20 epochs here. And we're doing the same thing below here as we did for the predictions earlier. And we're getting a 0.78 accuracy on this calculation, which is pretty cool, I think. And then we're going to do our kind of predictions and kind of look at our some of our false positives here if you want to check them out later. Where like a false positive here, it wasn't quite big enough, or a false negative, this was because of irregularity, things like that. So that is just, you can see some of the fits that they missed, and we can see here our and as a toy example, it's not perfect, but I think it's a pretty cool illustration. And look at our kind of confusion matrix, and we see our specificity is really good, right? If it's normal, we're really getting it almost every time. If it's a tumor, we're doing better than coin flip, but this is what we can improve on. And actually, it's the most important thing. If someone has cancer, you need to tell them they have cancer. So yeah, the model has a lot of room to improve, but I think you understand, the basic concept, what we're trying to do here. And you can see here, our sensitivity could be a lot better. It's only 0.63. It's better than nothing, but that would be room to improve. And here are some of the kind of filters and things we looked at, like the NN contu V learns filters. Kernel size is just your filter size. Padding is something you put around images, so they'll all be the same size. And then, pulling. Stride is when we can talk about. Stride is how many steps you move over, how fast do you filter over. Like if you had a higher stride, maybe you only would have gone across once instead of 3 times on one level. And here's the training as well, as we saw before. We see clear training. Valid's pretty jaggedy, but it still looks more similar to the training set. But you can see how the training set's so smooth. We probably need to work with it a little bit, obviously, to optimize our result there. And but just a couple of takeaways. CNNs, they look at spatial structure, right? That's what they know, how things interact. So nearby pixels are processed together. They're not independent like an MLP would look at it, right? Filters are learned, not designed. So the network learns what patterns to actually detect, right? And deeper networks, as you go to different layers, you can look for more complex different information there. And pulling allows us to also not only reduce dimensions, but also helps with translation and variance. For example, if there's a tumor left or right, it takes away like that should matter, right? And there's a lot of pre-trained models out there too. When you're doing things like digital pathology, a lot of people have made models. They're already radiology models like this. This is something a lot of you could more apply, but especially in digital pathology in our department. And so let's move to the next one, the recurrent neural networks. And so these are our, think about like the sequential things we talked about earlier, the longitudinal patient data. So, obviously in cancer research, we have one day of a lab result or scan is just a snapshot. But we need more information than that, right? So, recurrent neural networks and their specialized version, a long short-term memory, variance, exactly like readers. So they read the book, right, but they remember everything before, right? So they can distinguish meaningful long-term patterns, right, from the noise of different fluctuations. So you can see how, just in general, how this works with an RNN is it's fitting the model each week. Let's say we went through each week here. The first week, we would take the model, fit the model. It would learn a hidden state, pass that information down to the next step for the next week or the next lab sample. Do it all again, use that information to help with the next hidden state, on and on and on and on. You can actually make your final prediction and using what you've learned all the way down along the way, right? And kind of the math behind it, they actually use the TANH, the tangent H, and this kind of helps to improve the model. And you can see here just hand wavy. There's your WX1B. There's your model being fit. And then it's adding on the same fit from the -1 the last week, right? So it's going over and over, adding those on, and then finally into a prediction at the end, right? So RNNs really, they function by processing one time point at a time, and they maintain a hidden state that carries information forward. And along the while, they're kind of learning what I should remember or forget. But RNNs tend to remember, not do as good a job as they could at selecting what they should remember. And they also have a problem with vanishing gradients, which they forget early information. And that you don't always know which information they're going to forget. So this is where kind of the solution comes with the long short-term memory. So this is basically the same thing, except we introduce these gates in the middle here. So you can imagine you have their input, and the input's going into these gates, and then once you've gone through one time, the hidden state's also going into that, right? So imagine it's learning each time. So each time, the first time and each time in a subsequent time through, you're going to look into the first gate, and it's going to be like, okay, what new information is there? I already have all this old information. You got me some new information. What is important? What should I actually store this epoch, right? And then what should I discard? And then it kind of updates kind of along this cell state model as it goes. And then at each term, each time it outputs the answer, what should I output into the new hidden state. So it keeps going around these new hidden states while maintaining this kind of underlying state where it can kind of remember, right? And remember only the important things. And that way it can actually know what to discard from memory, know what new information to store, know what to output, blah, blah. And that allows it to be more efficient and solves the vanishing memory problem. So let's build one of these. So once again, in LUS, we're going to this time do torch stack to get our trajectory tensor. So now we're like a longitudinal tensor, and then we have our label for that tensor as well. Very similar before, we set up our data sets and our data loaders for our batches. And this is going to have 20 time points, 5 features. Set up our model. Once again, it's NN module. Kind of very similar stuff. This time it's NN LSTM function we're using. And it's going to have an input size, hidden size. And then we put that to another layer that's NN linear, which is your flattened, your fully connected, just normal output. And we're going to run this through just over and over and over again, keeping the same steps here. And then we'll output that last step as our answer. And same, our fit, exactly the same metrics, same optimizer, same loss function. Here we do input size of 5, hidden state 20, epochs 25. And let's look at the answer here, right? All right, so what happened here is it learned, this is a toy example, it was too easy, the data was too easy. So it learned really fast, learned as much as it could, then it didn't learn after that. You can see how the loss kept going down, but it didn't really help after this point. And same thing with the validation set, we got a very similar result. So what's happening here is the data is just too easy. The model's not messing up, it's a good model, it's doing well. The data itself is just too simple, and that is what is going wrong here. But like as a toy model to understanding, I think it works fine. I think it works perfectly fine for what it needs to be, right? So you have your input size, which is your number of input features, hidden size, the size of the hidden state, that memory capacity of that cell state that's remember that cell state that's being updated each time, that's your hidden size. And your number of layers, how many times do you want to make just sequential LSTM entire stacked heads, right? So you're basically stacking entire models of these learning cell states, one after the other for the number of layers. That gives you obviously a deeper network. Don't worry about batch first, it's just a formatting thing. And the dropout is the same as before for the regularization. And we're gonna do our predictions here, and we're gonna see they're really awesome as we'd expect. It's a little bit too easy of data set. But we still have, I think you still understand what is happening here. And if we think back at that graph we saw, right? Remember how the responder just came down, and then it's always like, what did I do this on the camera? Remember the responder came down, right? But that the delayed response, the relapse came down then back up, right? Or you know what I mean? As a U, right? So that responder, that blue one, showed just a clear, obvious downward trend. I mean, even a simple regression could have caught this. We didn't need a fancy model. However, the non-responder is kind of weird. Like for the first 10 weeks, he looked exactly like a responder. But there was a sharp U-turn, and if you only looked at the first half, you would misclassify him. So you need longitudinal information the whole way, right? So you need that when this trend reverses, and LSTM moving along is going to remember what it looked like before, so it's going to capture this U-turn, right? And most importantly, like once we have a well-trained model, we can try to find when that actual momentum change happens early on, so we can catch it early, which is the most important part of I think this modeling is to catch a non-responder early on, right? And like we talked about, this is a toy model, and it seemed obvious. This guy was higher with its deal, but remember, think of thousands of patients, think of multiple features each week of our lab results. Think of some labs going up and down. It's going to be very hard to pick that out and be very complicated, you can imagine, to be able to find that with a simple model. It really helps to have the memory as you go along the way, right? And one more, I want to show you another Homer problem. I did that as well too. I think it's interesting. Is this guy? Imagine the same thing. We have longitudinal data, and we're trying to see if a treatment worked or not. And this is a really great example where we start low, treatment's working, and then we relapse, right? We just end up right before we started with our white blood cell count here. But we also have guys have a delayed response. Things got worse, but then they finally responded, and now they're doing a lot better. But the data is exactly the same before too. So like if a random forest, some models look at this, they're gonna be like, okay, we see all the data, we see the variance, standard deviation, all that stuff, right? And they're going to say like, oh, they're more likely going to find a problem here and be like, oh, we don't see this. Since you're not looking at it sequentially like an LSTM, you wouldn't necessarily know which way this flipped. You would just see the data before and after, and you wouldn't be able to tell between these two things. So that's kind of another example I think is pretty cool to show how that works. So let's look at our transformers now. This is actually really cool, and it's a kind of an improvement over the RNN and LSTM. And really, this is the self-attention revolution. Things like ChatGPT, single cell BERT, all these really big new AI models. A lot of them use transformers, and self-attention is this big thing that's important. That is giving them the new advantage over the RNN and LSTMs. So think about this. When we use the RNN or the LSTM, remember, it read data like a sentence. It went word to word to word from left to right. Each time it read something, it had to do all its calculations again. Well, that's slow, right? And you're not getting all the information at one time, right? And you may even forget or lose some. Well, transformers utilize actually attention to look at the entire sequence at once. So you can only imagine this makes them significantly faster and better at understanding all of the concepts, right? All the context, how it all works together. So think about it as attention mechanisms like a highlighter. So these guys looked at instead of reading the book line by line, it looks at the whole book at once, right? And then it doesn't really care about remembering everything in order. It's just like, what is important in this book? You know what I mean? And it looks at everything and finds all the most important things that led to the, let's say the novel was an Agatha Christie novel. It would find all the best clues for Poirot at the end to decide if you're the murderer or not, right? So it's not going to remember every little detail. It's too much. Plus, it's going to be noise, right? It's going to hurt the thing. So it's going to look at the entire thing at once, find the important information, and just work with that. And you can imagine how this is better. Well, one, you can parallelize them. They don't have to wait for step one to finish to go to step two. They do everything at once. Looking at everything at once, they don't have finishing memory. They don't forget things. And the global context, right? They can look at how long-range relationships fit together better, right? And here's kind of an example here where you can see that we have the 20-week things. We just have, like, let's say 5 different sampling points, and week 1 gets the highest attention because it's a baseline matter. It's a baseline prediction, right? It's going to really matter to see the difference. The last one's really important, but it could be, who knows what the data is. The point is, it's looking over everything and deciding which percentage of each week is important to include. And within those, it's probably choosing which genes are important, right? Or which, features are important. And then it puts all this into a weighted output, basically, that combines all the positions based on their relevance. So, the idea here is self-attention learns relationships between all the positions in sequence, right, at the same time. Now, the key thing here are these 3 matrices: a query matrix, key matrix, and a value matrix, okay? So your query is, you're querying. You're basically saying, what am I looking for? So you're setting up a parameters of what do I want to look for? So that's basically, send your matrix, what do I find? What am I looking for? And then the key matrix comes back and it's like, okay, what do I have? Okay, here's what I'm looking for, okay. And it looks at everything at once, and it's like, but then what do I have? Okay. What am I actually finding? What do I have out of what I'm looking for? And then the value matrix is, what can I actually contribute? Now that I know this information, what can I contribute to help make this prediction down the store, right? And along the way, you have these attention scores that kind of just tell you how much you should attend, right? How much should you care about certain relationships? And then you have multi-head attention, which is really awesome, that you can have different heads at the same time. And different heads can be looking for different things. And they can all work at the same time, parallelized, right? So it's like super fast. You have 100 heads that do it at the same time. And you have positional encoding, which it tells the model about position along the whole way. So you get the math. I'll kind of hand wave it because I don't want you to, it's a little bit complicated, but you basically have your query key value matrices, and you basically set them up and work through them, and you finally get a softmax prediction. We won't go through that. It's kind of complicated. And so just thinking about this, I think this is the way of looking at this is, you have your input here. all positions go into each head at the same time. You have different heads that are run through their query key value matrices and provide an output based on whatever we want. So the first one's just looking at recent trends. Second one is calculating our baseline. Third one, we're trying to find sudden changes. Last one, we're looking at periodic patterns. So you can imagine how well you could have adapted this. Remember that non-responder delay responder? You could really adapt this to look for exactly what you want, like early momentum breaks, things like that. So it's just a really in-depth LSTM, right? It's much faster and better. So each of these guys makes their prediction, right? And they output it all here, and we concatenate all these outputs and then run them through our final activation function to get the output. But by concatenating all these different results, we're getting a much more powerful, robust solution, right? So just to go over it one more time, you have the parallel processing, same input sequence goes in each one, the separate projections or calculations for each head, and where each one learns a specialized pattern, and then we pull them all together for our final projection here. And what we're going to do, we're going to build one of these. So this is really hard. You would never try to build one of these in R, and don't worry about the math. The program once again, but it's kind of similar. And in module, then I needed an embedding and an encoding deal here. So I need to embed to these hidden ambitions, and I need to give it the positional encoding to start off with, and then I can run through my different matrices. So for each head, I'm going to have my query here, and my key, and my value. So I use the eliminator for that, and then my outputs are going to be my fully connected, right, with dropout and some normalization. And that's basically it. And we run it through. It's kind of complicated. Don't worry about it. We find our attention scores, apply those, and attention scores help make the output for each one of the heads. Blah blah. It's kind of complicated. Don't worry about it. Attention models are fit. This is the same laws, same atom, same metrics. So all of this is pretty much the same. We changed the hidden sizes a little bit to what we wanted. Epic's 50 here. And make our prediction very similar as before. And we have a very good 0.93, 0.97 here, 0.97 ROC. Once again, it's an easy example, right? You saw how good the LSTM worked. So toy example, really easy, but I hope you understand, what we're trying to find here. And so it's really just managing this query, this key, this value, I think. Really understanding how these work together. What am I looking for? What do I have? What can I contribute? That really is a high-level understanding of how it works. And it's really just measuring along the way, it's basically measuring this attention as it moves along, right? To be able to understand how relevant different positions are together. And it has, that great softmax output. And that's basically it. And if you plot the training, it's obviously really, really good, kind of like LSTM. But I think you see the examples here, the multiple attention heads, the parallelization, multiple layers, all those things make it, and you can see why it's an improved version that everyone's so happy about. Now, this is a really, really simple model I made. It's the best I can do in R. It could have been a little bit better, but anyway, we only had one attention head, one layer, and a basic feed-forward network because I didn't want to get too lost. If you wanted to look through the code yourself, I didn't want you to get totally overwhelmed. Where a full transformer would have like 8 to 16 multi-heads at the same time, each one looking for something different. You would have different layers, and all this is much better, and you would do it in Python. And that's where you're going to get things like natural language processing, GPT, BERT. Those are all things you can work on in Python that we're going to try to do in our next workshops. We compare the accuracy. They're very similar between the two because the data was so simple, you really weren't able to see the difference. But much more difficult data, I would expect self-attention to do better. And there's actually a homework problem that kind of goes over that at different links here. Or I think a practice exercise, actually. And here we kind of already went over all this stuff. If you want to look again at some of these explanations, it's a little bit clearer. And then once again here, you can't really use R for a lot of the stuff. For some of the best functions we need that work in PyTorch aren't transferable. But for teaching, I think it works really great, or playing around. All right, so our final topic we're going to cover is graph neural networks. And this is a learning by association. And These can only be done in Python. There's no way to make it one in R of any value. So this is going to be super conceptual. Just I want you to understand, because these are becoming some of the most powerful models and something we'll definitely work on later. And just think about this. Biological entities rarely act alone, right? They're all connected, extremely connected. And like a mutated gene is only dangerous because of how it influences everyone around it. If you have a mutated gene, it doesn't matter. How does it influence the other genes in the microenvironment? And gene editing can really design to respect and find this connectiveness. And the core concept of this is message passing, which you look below here, kind of this protein interaction network with TP3 in the center and these different genes it works with. It's really a gene interaction network in a way, but the core concept really, remember, we look think about you're at a party, and you always love my party click explanations. Why is it always a party? We're at a party, we're at a gathering, and each node just kind of looks at its intermediate people, right? And it listens to them. What are they saying? And then it pulls in that data into itself, and it updates what it knows based on the gossip they're hearing, right? So after a few rounds of this, it kind of knows what's going on around it and everyone's nonsense that's happening, right? Everyone's gossip, it knows. So then if you asked it about something, it would know exactly what to say about so-and-so, right? So this is kind of the idea here. We make this graph of a network. For example, we made a graph here. You see there's different genes have different contributions. They have different weights. So you can see here the thickness of the lines. And they just all connect in different ways, right? And basically we're sending the features. We're saying we're message passing, and it should be back and forth, really. We're message passing all of TP3 and among each other and around to update our understanding of this whole thing. So by looking at the different connections, we have an idea, a graph of what is happening in the specific thing, a specific pathway. So you can imagine you could set up a graph for each patient, and it would have a different representation of this exact network. And it's very possible that you could also make a prediction out of that and say, well, if they have this type of graph, then we know they're in that stage, or we know they're going to metastasize, or we know they're, have a certain level of disease, whatever. Or you can do things like let's say viruses, like people make phylogenetic trees out of viruses. That's just a graph, right? And they look at the different trees, and they're able to tell what type of virus that actually is. And another way of looking at it is the drug discovery. You look at thinking about a chemistry. Chemistry looks like, think of a carbon atom or a nitrogen atom, or the way, remember old chemistry atoms. The bonds can be the node. The element is the node. The branches are the edges are the bonds. You know what I mean? And you can look at the representation, make a graph out of different molecules, and see if they work associated with different drugs and different outcomes. The point is, you can make a graph out of anything, and it's just an interconnected, basically just an interconnection of message that is shared between it. It could be directional, unidirectional. Like a tree would be unidirectional. This here would be multidirectional. And when you basically make this graph, learn all this information, then you can obviously make predictions on these different graphs because they are different. So just looking at the math behind them, don't worry too much about it. You're kind of just, each node starts with its own features. It sends messages back and forth to their neighbors. It learns each node, like each node in the tree, for example, would learn these new information, and it would pass information down the tree to each different branch, right? And then you would have kind of this aggregated understanding of each node that would set the entire graph up to be unique. Then you could look into that graph to have information or use it as prediction, right? And really, we talked about MLPs best for tab, CNNs best for images, RNN best for sequences. Well, this would be GNN's best for graphs. Obviously, it's a graph neural network, right? But graphs are a very good way of collecting information spatially or sequentially or just among different things that have a lot of interaction with each other. And to me, it's the highest level of this. So, for instance, you can make a graph transformer, and that has position encoding that's unidirectional, for example, on a tree. So I think this is kind of the highest level. Look, moving toward GNNs, graphs, things like that. They're adding on a lot of detail to more complex problems other than just text. You know what I mean? And like when you go to Python, you're going to be using PyTorch geometric or GGL possibly. There's also TensorFlow you can use, but TensorFlow was made by Google. There's a joke that TensorFlow was made for computers, PyTorch was made for people. So TensorFlow is kind of complicated to use. It doesn't have as much, a big a community, but unless you work for Google. But it's really great, and it's a really great method. It's just as good as PyTorch. PyTorch is a little bit simple to learn if you're a beginner. And you can kind of see how this would work here. This is one PyTorch example here that we would do in Python this is actually a graph attention network. And you can see how it's very similar. The initialization was kind of similar. Remember that the self.convo 1 and 2, those are similar. So we're doing a convolution actually here. And we're doing a graph convolutional network. And then we have the self-forward, the different layers, the ReLU. So it's actually not that different, right? So just having seen this before, hopefully, if you just play around with it too a little bit, once we move on to that, you'll be able to really see the advantage of it, right? So GNN, they're really good at looking at relationships between entities and the data, message passing, obviously, and you can have multiple layers and multiple distances to choose how many neighbors, all that stuff. And it's really, you can see here, spatial transcriptomic pathway drug. These are at the very cutting edge of what we're using today. And if you look at all those models just side by side, what happened here? Well, just at the accuracies, and we see here, that if you remember back, the sequences, the image, and the tabular data, and we kind of talked about that before, how these all kind of related to each other and kind of explained, and how they worked. And here's kind of an architecture selection guide, what kind of data you had, what you should use. We already talked about that a little bit. Just more examples on that. And just to run you through a couple really simple, I can give you some practice exercises at the end. You don't have to actually run through these all because the code is complicated, and how do you know how to do it? I didn't teach you. Just look at these as conceptual examples here. So the first one, all we're doing is just tuning hyperparameters. So I just want you to show you that you can actually tune these things. They're just harder, right? So we made some model just like above, very simple model. It just has one layer, ReLU, dropout. It's just has an input to hidden to hidden, and then one output, so 3 layers. And very simple. You basically set up a search grid, just like before, for tidy, and then you run it. This function goes through. You set up your data sets, data loader, and then we fit the same way, except we're running, we're tuning that grid along the way, like here, this HP grid, this tune. And this one, man, this took forever. And once again, I did a multi-site, I set up parallel for it, and it still took forever, like a long, long time. Anyway, but I just want to show you that you can train these models. I tried 81 different models here. And I found the best one here, and then, plotted the result here, and you can kind of see how, you can kind of step through this and see how, since in a higher learning weight did a lot better here, did a lot worse here, lower learning weight worked better. You can kind of flip through this and just kind of see how the penalties and the network width kind of helped out. Like you can see, it's not really bigger is not necessarily better in this instance. So you can see here, things like, here's all the final, here's all the final results and kind of an interpretation of some of the results here. But a low running rate and model doesn't have to be the greater than it needs to be are two really important things. And then I work through a second problem where I just kind of go through and I look at the same problem we did earlier. We had the 2-layer, and I build a 1-layer and a 3-layer, and we just kind of see, like, as we go through each one of these layers, are we getting more information? How do these stack up, and how can we compare to them? And same image data. I don't really want to go through anything but the results here. You can take a look at that if you'd like. One thing I did was I talked about batch normalization, which it just kind of stabilizes the batches where each layer's outputs can have different scales. So these are normalized and makes the training more stable. This allows for a lot of things like faster training and less. It's kind of a little bit regularization. Anyway, let's look at the results here. So our low complexity model, just one filter of 32 neurons, didn't do. This is our first one. The result's not here. The second one goes 32 to 64, and you can see it gets smaller. Remember, it was 28 to start. So this one half twice. And this one we put 16 to 32 to 64. And we ended up the final image size from 2828 to 33. Same with the batch normalization. And we just checked out the results here. We trained them and we got the results. Let's just compare them. And what we see here is the one layer did not do that great. We needed at least 2 layers to get a good result. Batch normalization didn't really help that much, but the third layer helped as well, but not as much, right? So I don't even know if these are statistically different, right? So the idea is you go higher if you get a result, but you're instantly going to hit a level where you don't need a higher level. And it's kind of looking at the training curves, so you want to check them out, and kind of some of the clinical consequences of these different metrics. You can kind of see how the specificity and the sensitivity is going through these. So let's go to the third one here. And third one, as I talked about earlier, I did LSTM versus self-attention, but I did it at different sequences. So we started like small. It's the same deal. I'll show you when I actually train it. So you can see here, I did short. I did a 5 time points, 10 time points, 20, 40, 60, 80, 100. And I wanted to see if one worked better than the other. And let's look at the results. And what we're going to find is attention needs data. So let's look at the accuracy by sequence length. So as we go up in sequence length here, you will see LSTM jumped up really fast and got good accuracy, but then kind of tailed off at the end. Attention kind of started slow, and as it started learning, it started going up a lot more. And the idea is if we had a lot more sequences, I bet it would surpass it, especially if we tune the model a little bit better. So we see that LSTM is just real. I mean, self-attention or any kind of attention needs a lot of data. It's very data hungry. But one good thing about it is it doesn't, as you add more data, it doesn't need more speed. So LSTM continues to go up, and I'll show you here. LSTM just continues to go up with data. So, I mean, we don't have this, even if we built Elon's data centers on Mars, it would still keep going up, right? So we know that attention is much better computationally, and if we go high enough, it probably ends up catching it. Because neural networks are really good in the long run, as we showed, right? So anyway, that is kind of our ending. If you go down to the bottom here, I gave you some just good summary of different things we covered, some different practical points that help you out, decision frameworks, some quick reference guides. I know this is kind of overkill with all this stuff, but who knows? You might want a chance to look through some of it and give you a little bit better description of some of the things. Here's just a basic LUZ model template if you wanted to play around with some of those models. You could just change up the ones I did a little bit. And then I made some additional assignments that are really pretty cool. The first one I showed that I did a multi-class prediction. Let's see, and then I did for the second one, I did the sample size one I showed you the result on. Then I did one where I did not show you on, but I used even a more complex, different architectures of CNN to see if we got an advantage with a more intricate model. And then finally, I did that sequence showdown at the end that I showed you as well too. And at the bottom, I have some kind of additional resources, things you can look at. This is kind of an idea of when we're going to move to Python, and that's it. We're done. That's the end of our really 15-part series. If you guys say everything from the beginning, you've seen my beard grow a lot longer. Santa beard for Christmas. But you work through the R, work through the stats, work through the machine learning. Proud of you guys. You stayed along the whole way. I hope it really helped you out. Don't be discouraged if you're a beginner. Honestly, if you could get through the first 2 parts, the basics and the stats, I would be really, really happy and really proud of you. And just grow into the machine learning. You don't have to do everything at once. I just wanted to show you everything. So in the future, we can just apply these methods. So go through things, build up slowly. You can always go back. The whole idea was to distill all this information into one place for you that had a linear progression. So you would have someone that guide you in a linear progression to get where you need. And if you start from the beginning of this and you go to the end, you'll have that. If you're already an expert, start machine learning. Learn some new stuff about that too. But you can do it. Keep going at it. Computational stuff is important. No reason to be scared. Keep learning. And I hope to see you later on for some more sessions. Thanks for attending.